Some bacteria are harmful, for example, H. pylori. H. pylori is a type of bacteria that resides in the stomach and infects more than 50% of individuals worldwide. I know what you're thinking. If the bacteria does not infect everyone, then what makes it so special? What makes this bacterium so unique is that once the bacteria infect the stomach, some people do not experience any symptoms, while others develop symptoms that can be so severe that they lead to gastric cancer. The overall research question was to investigate and determine why and how an individual's health can change from asymptomatic to symptomatic. I'm participating in a study conducted by the US National Cancer Institute, and 700 samples were collected worldwide. This is the largest study in the world on this topic. The aim was to focus on the H. pylori genome in order to find further evidence showing why this occurs. Since the H. pylori genome is more than 1.5 million base pairs or letters in length, my aim was to only focus on one section of the genome called HOPQ. HOPQ is a gene that encodes an outer membrane protein on the surface of H. pylori. HOPQ has been one critical component of the H. pylori genome that is known to have the biggest association with gastric cancer. Due to this, I looked at the relationship between HOPQ DNA sequences and the genetic ancestry from the samples collected. I created a phylogenetic tree as a visual representation of the evolutionary relationships amongst HOPQ sequences and established categories based on each grouping. My goal was to determine whether sequences that led to the development of cancer grouped together or not. If groupings were found, this would suggest that something specific within these sequences was shared. This could shed light on HOPQ's role in gastric cancer, which is exactly what I found after grouping the sequences based on the tree. I would like to draw your attention to the figure under the analysis heading. You will notice groupings denoted by different colors. The largest grouping is highlighted in red, and by a regression analysis contains significantly more sequences that led to gastric cancer than to asymptomatic outcomes. These findings were pivotal to our project and point us in the direction to take next. In terms of future research, I really look forward to further examining the specific DNA sequences and the strains that showed the highest association with gastric cancer occurrence. Thank you.